about the bovine hemoglobin adsorption at pristine water-air interface. And the second part deal with the muscle-inspired imprinted polymer surfaces for the protein sensing. All right. So now comes the second word which I have used in the title of my talk was the nonlinear spectroscopy. But before I will go into the detail of nonlinear spectroscopy, I would like to start with the very basic one, the information which we normally study in our 10+1 grade, right? So let's say that we are having one incident field which is of the low intensity. So we all know that it kind of induces the dipoles in the materials. And we can write down the expression of polarization vector P in terms of susceptibility, this epsilon naught, and also the electric field intensity in here. So that I am designating as P1, which is the first order one, where this sky is the linear susceptibility. All right, and let me just, for the sake of completeness, say that the electrical susceptibility of any material is a measure that how easily any material can polarizes in response to the applied electric field. So we have studied that for low intensities, this polarization vector is linearly varying with the electric field. So far, so good. Now, we come to the very high intensity field. That is, when I say that very high intensity electric field, what I mean is that the electric field which we are going to apply to any material is going to be the order of the atomic electric field strength. So that should be roughly of the order of 5 into 10 to the power 11 volt per meter. So similarly, we can write down the expression of mu, or let's say the polarization vector, which we can clearly see is not anymore a linear function of applied electric field, right? So this material response actually gets perturbed and become a non-linear with respect to the incident optical field. The optical properties get perturbed to certain extent, and this optical response becomes non-linear with respect to the incident field dependency. And that's why we express this polarization in terms of the perturbed series with the and second is chi1 in here, and the chi1 is the first order susceptibility, which we have defined earlier. Chi2 and chi3 are the second and third order nonlinear susceptibility. All right. So now coming to the nonlinear optics. We know that nonlinearity exists when we apply the high intensity electric field. Let's say that we are having one source and such high intensity electric field can't be taken from the laser sources. Let's say that we are having one laser source, right? And in the presence of one laser, it will lead to the generation of the second harmonic. You can clearly see in here that the polarization vector term is expressed in here, and we see that there is a generation of the second harmonic. When I say second harmonic, that it means the outcoming radiation is going to have the twice the frequency of the incoming one. Okay, fine. And if let's say that if we shine a material with two incident fields, so this is some kind of material in here, and we are shining this material with two laser fields. So one of them is having a frequency of omega 1, another one is having a frequency of omega 2. Little bit of mathematics and you will reach to the final term in here, which has different terms in. And you can clearly see that one of the term is the sum of the frequency of the two incoming lights in here. So that is what is responsible for the generation of the sum frequency signal. And if you see in here, there is also a generation of different frequency, but that is not of importance for the current talk which I'm going in here. So to summarize, this is the Jablonowski diagram, or we say in simple word, energy level diagram where we have two input lasers, which gives us the one output laser beam, which is having the frequency sum of the two incoming laser fields. All right. So now coming to the some frequency generation spectroscopy. This is the general schematic setup of the this technique, which consists of one laser source in here, and the setup which I have used, the ND-arc laser was used, which is having a wavelength of 1064 nanometer, and it is being fed into the second harmonic generation unit, and after that we get three different kind of beams in here. Since it's being fed into the second harmonic generation beam, now the outgoing beam, which is the green light in here, which is visible light, so the twice the frequency of 1064 nanometer is going to be 532 nanometer. So these kind of radiations we get after the second harmonic generation unit. And this red light is the original 1064 nanometer in here. So these three beams are being fed into the optical parametric generation and difference frequency generation units, which actually gives us again two beams. One is the visible beam in here, another one is the tunable IR beam. So this tunable IR beam is having frequencies which lies with the ranges of the, which can match with the molecular vibration of the molecular intensities which are present at the interfaces. So to summarize it up, SHG occurs when two pulsed laser beams, one of fixed frequency, which is the visible one, and the other is the tunable IR in here. They achieve spatial and temporal overlap. So there has to be both kind of overlap at an interface in here. The light is emitted at the sum of the two incident frequencies. So this omega SFG is going to be sum of omega visible and omega IR. So this is where the energy conservation condition takes place, or it holds true. The intensity of light is resonantly enhanced when the frequency of the tunable IR beam, this beam in here, is coinciding with the vibrational mode of the molecules. We know that if we are having some molecules, they have some kind of intrinsic molecular vibrational frequencies. So there will be a resonance or enhancement in the signal when this frequency of tunable IR is going to match with the molecular vibration of the molecules present at the interface. So by detecting this some frequency light, this we mean here, as a function of infrared light or infrared frequency, a vibrational spectrum, which is here, is obtained, which is upshifted into the visible region of the electromagnetic spectrum. So this is the whole purpose of using this fixed frequency wavelength or frequency in here. All right. In order to achieve the some frequency generation, the spatial and temporal overlap has to be done. So it is essential condition. A coherent some frequency signal is subsequently generated at an angle. If you see in here, if we know the angle of the incident beam, we can calculate the angle at which we are going to have this some frequency generated signal in here, simply by using the phase matching condition, or we say the conservation of momentum condition, right? Or for all these three beams parallel to the interface. So the intensity of this some frequency generated light is actually directly proportional to the susceptibility, which I already described previously in previous slides, and it's also a function of the intensity of visible light and the IR light. So to discuss that, why I am so much concerned about, or why I am representing this specific technique, I will in a minute show you that how this technique is quite distinct from the other conventional spectroscopic uh, tools. So let's say we might already know about the Fourier transform infrared spectroscopy, right? So what you do is that you take a sample, let's say it's an ice sample. When we say ice, it means we already know that the water molecules are tetrahedrally bonded with their neighboring atoms in a ordered form. That's why we call it as ice, right? So if we put this sample under normal FTIR, what we get is some kind of hump coming in here, which is the characteristic of the uh, highly coordinated water molecules. Now, if we change the sample and we put liquid water, right, it is rather having a very huge hump and very broad one in here. So in addition to 3200, we get a high peak in here at 3400, which is the characteristic of the, or it is a signature of the tetrahedral coordination. This one is a tetrahedral coordination, and this one is the less ordered molecules around 3400. All right. So instead, if we take this liquid sample and we put it under the probe of some frequency generation spectroscopy, what will happen that we will have some kind of this spectrum in here, which is representing in here. So we get not only the ordered OH species representing at 3200 and less ordered one, which are not tetrahedrally bonded one at 3400. In addition, we get a very sharp peak, which is coming out to be at 3700 centimeter inverse.
Naveen Markan.
Hello?
Excuse me, ma'am. Naveen Markan, please stop presenting your screen.
Shall I
Please
continue?
continue. Yeah, please continue.
Yeah. So we are having this sharp feature, which is coming at around, let's say, 3700 centimeter inverse. This corresponds to the free or dangling hydroxyl species. Right. In here, when we proved it using the FTIR, we had some kind of feature coming in here, but it was not really resolvable. It was actually taken. It was dominated by the signal from the bulk material. Whereas in case of SSG, we clearly can see this free OH species, which is coming exclusively from the interfaces. To understand it schematically, you can see in here the below subsurface, or like below after 2 or 3 layers from the air, this all water molecules we term as the bulk water molecules, and these molecules which are above the subsurface here, they are surface water molecules. So some of them are tetrahedrally bonded, which is coming in here at low wave number. Some of them are coming at in here, which are not tetrahedrally bonded, but are having two bonds, hydrogen bonds with their neighboring atoms.
मैडम,
Yes.
आपकी स्क्रीन प्रेजेंट नहीं हो रही है, 5 मिनट से स्क्रीन नहीं आ रहा, केवल आवाज आ रही है.
Okay.
Actually, मैम किसी ने बीच में स्क्रीन शेयर कर दिया, तो that is why.
Sure.
Yeah.
I will try to. Yeah.
Or maybe you can try to pin your screen. The participant who asked the question, all of you could pin ma'am's screen to your full screen. That's what I did. I thought it would be working for everyone.
Fine. By the way, I saw that this meeting is being recorded. Is it being recorded by the organizers, or some of the participants are recording it?
Yeah, yes, ma'am. From the by the organizers. Shall we do that, or shall we stop that? Like, are you okay with that?
That should be fine as far as it stays with you.
Okay, thank you.
So let me share the screen again. Can you see my screen? Now?
Not visible yet. Yeah, it's going.
Now it is fine.
Yeah, ma'am, fine.
Good. So I was explaining this. Yeah. I was talking that shall I repeat because my screen wasn't visible before. So I was making sure that you understand why I am talking about. I have some kind of echo. Hello,
Hello.
hello. Yeah, now it's fine. Okay, great. So what I was saying that under any normal IR spectroscopy, you won't be able to observe these free OH species present at the interfaces here. And this is the pictorial view, this is the schematic view, how you can imagine that this is water molecule here, and this bond is actually really at the interfaces. Can detect it using the computer generation spectroscopy. So, in summary, unique feature about BSH is that it is surface and reflective. And since it very much lies on asymmetrical behavior of molecules present at interface, we won't be having anything coming from the bulk of it. So in a way, ground free technique. And this is being used for the chemical applications, and it is also not this technique. Okay, so now coming to the first part or the first project I want to present in here is the bovine globin adsorption at Princeton water air interface. So, to start with, what is hemoglobin? You know about the blood cells, some kind of representation is given in here. So, if we see the hemoglobin in blood cells, we see that actually,
Hello, Dr. Shilpi. Your voice is not clear. Some disturbance is there, some kind of disturbance. If you can check your
uh,
mic.
is it now?
Yeah.
It's little better.
No, no, still some disturbance is there.
मैडम आपने दो जगह से क्या है वो लॉगिन
Then I'm
Or
login
can we
with
ask some
yeah.
outside particle if they are hearing it properly, because it may be due to reason of slight internet?
नहीं
Yeah,
मैम
it,
नहीं.
Because Shakun has written, so she is also here. So can we ask from someone which is not from slide that is the voice clear or not? No one is responding.
नहीं मैम प्रॉब्लम है.
Okay,
what is this
voice is
or
breaking here.
थोड़ा इको हो रहा है मैडम
I also thought, I mean, I can also hear a lot of you. How
आप
I
आप
leave the meeting and rejoin them, or it's somewhat
मैडम आप
sometimes
जो
it's a
थोड़ा आप
yes,
फिर से ज्वाइन करो आप लॉग आउट करके लॉग इन करो
person.
लॉग आउट करके लॉग इन करो फिर से
Hello.
हेलो.
Now, yes. I'm speaking now. So, is there still persistence of this echo?
No, ma'am,
Yeah.
it looks better now. It sounds better now. Hello?
Hello. Just give
Shilpi,
me a
you say
moment.
something?
माइक. हेलो शिल्पी, ये माइक की तो प्रॉब्लम नहीं आ रही है. Due to some internet problem, Dr. Shilpi has left, but she will join immediately, but still will.
Am I audible clearly now?
Yeah, Dr.
Yes,
Shilpi.
ma'am. Yeah.
Hello.
Yes,
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 ma'am.
Dr. Shilpi. Okay.
Great.
share your screen.
Ma'am, could you please start from your previous slide because that was not audible actually.
Sure, sure. So this was the last slide, and I guess I made sure that the participants can relate, or they can actually realize the importance of vibrational frequency generation spectroscopy if they compare it with using the different conventional spectroscopic tools. I mentioned about that how VSFG is surface sensitive and can be used for the chemical identification. And since there is no contribution comes from the bulk of the material, which is the very principle for the surface frequency, some frequency generation spectroscopy, is because of the asymmetrical environment. Right? So it is background free technique, and also this is non-destructive technique for the samples. Anyway, so now let me come to the first part, which is the first project I'm going to present as an example that how we can prove the uh, absorption of bovine hemoglobin at pristine air-water interface or water-air interface. So I was here and I was talking about the bovine hemoglobin, which we selected as a model protein molecule to probe its molecular structure and interaction dynamics upon adsorption and its influence on water structure at the air-water interface. So these are the red blood cells in here. I'm sure that most of the participants might be familiar. And if we see in here, this contains 4 polypeptide subunits. 2 of them are characterized as alpha units. 2 of them, rest of them are characterized as or known as the beta subunits. Each of them are having a heme bearing group in here. There is nothing, this is not about the biology. If we try to understand it in terms of the physical chemistry, then we can see that there are some kind of organic molecules present in here. So they will definitely give us the vibrational signals. Anyway, so this is the slide where I am representing the result. So this figure in here is showing us the OH stretch region when we place our sample under the probe of SFG. So what we have done is that we started mixing hemoglobin in water itself. And we placed our sample in front of the SFG probe. So the dominant sharp peak, which we already discussed when I discussed the water spectrum, is nothing but the free OH peak. The peak at lower frequency in here, somewhere here, is coming from the tetrahydrally bonded water molecules. And in this broad spectrum, a peak is also appearing in here, which I am assigning it for the weakly hydrogen species. Right. So you can see that for lower concentration, not much is happening in here. It has been observed that with the increase in the bulk concentrations, when we are increasing in here, the arrow mark represent in this figure, which is here, shows the change in the hydrogen bonding environment. So you can clearly see that starting from the pristine water, which is in black color here, we see that this peak is getting diminished when we are increasing the concentration of hemoglobin molecule. And if it's not clear in here, what I have done is that I have taken the integrated intensity under this peak, and we can clearly see that both the free OH peak has been gone down significantly, and also the peak which was coming from the less coordinated water molecule is also coming down. So definitely by adding the hemoglobin molecules, the interface of air and water is getting changed. Now, let me go back to the other slide first. Yeah, so this one in here. This is the CH spectrum. The previous one which I showed was the OH spectrum, which was showing the change in the water molecules. In here, if we see, this is the molecular conformation of the bovine hemoglobin. So it clearly is showing that these hemoglobin molecules are present. So if we see in here, for couple of concentration like 1 p.m., 5 p.m. and 10 p.m., they are not in here. The molecular conformation we cannot confirm because the concentration was so low. But from the 30 p.m. and onwards to the highest concentration, we can clearly see the peak coming in here, which are the fingerprint regions of the hemoglobin molecules. So the peak in here, this is CH3S, is the symmetric stretch which coming out from the hemoglobin molecule, right? Since I have already shown you the this. Hello. Shall I continue? Is there any problem?
Yes, ma'am. Please continue, ma'am.
Okay, fine. So I was talking about the hemoglobin molecule. So since it has both alpha and beta chain, and they consist of aliphatic amino acid and almost 400 methyl groups, so which are presented in here. We can see the molecular conformation. And it also has 50 aromatic residues in both the chains. So the aromatic stretch is quite visible. So we have absorption of bovine hemoglobin molecule as a function of its concentration.
Excuse me, ma'am.
Yes.
Subita ma'am, please mute your microphone.
Yeah.
Okay.
So the
Please
insect,
continue.
the insect which I'm representing in here, it shows the intensity variation of this methyl species in here with an increase in the protein concentration. The increase in SHG intensity of this methyl stretching mode does not reflected as the estimated surface coverage, corresponds to the variation from low to high concentrations. It has been reported in the literature at various places for different protein molecules that the surface coverage increases to nearly 5 times by increasing the bulk hemoglobin concentration. So we should have some kind of surface coverage for the different concentrations. However, in the present scenario, the SFG intensity which we obtained for the 800 parts per million protein bulk concentration, if we calculate the surface estimate, it is coming out to be just 2 times in comparison to the intensity found for the 5 ppm. So the surface coverage is not that much as we were expecting as per the concentration of the hemoglobin molecules in here. So the observed intensity for high concentration in comparison to the low concentration suggests that the protein molecules may have unfolded significantly at low concentrations. in comparison to the high concentration. In fact, at low concentration, the protein molecules at the surface have enough spatial opportunity. So what happens is that at low concentration, they will try to migrate towards the interface, and then they will have lot of space to spread around. And since there is a presence of electronegative entities in the air, the protein molecules will start to unfold, and they will spread on the interface. But in the case of high concentration, the unfolding becomes limited by the dominant presence of protein-protein interaction. Rather, they won't go for the unfolding up to the same degree as it can be for the low concentration. Fine. Now, this is the steady spectra. If we see in here, for the, let's say, 5 ppm spectrum, we see what we have done is that we waited for the spectra to achieve. Let's say the first spectrum was achieved at 0 minute, and then we have taken roughly after half an hour every measurement, and we clearly see that some dynamical changes are happening without even changing the concentration. So this clearly suggests that there is some kind of diffusion going on, and which is after diffusion, there is unfolding of protein molecules. All right, at low protein bulk concentration, it takes time to reach to the equilibrium. So you see that after 84 minutes, I am not showing the spectrum in here, the spectrum does not change anymore. The steady state measurements, which are in here, for both the concentration, you can clearly see the time difference. So after half an hour, for 5 ppm, there was not much of difference. For 56 minutes, yeah, we started to see some kind of change in the OH spectrum. But at 84 minutes, we saw the spectrum got really changed. In case of 10 ppm, we clearly saw that after half an hour also, the changes were very much vigilant in the spectrum, and they were very significant. So in low concentration, the changes happen after a while, whereas at higher concentration, the changes happen very facile. So the steady state measurement for both the concentrations, they reveal similar spectral changes in OH scratch region at different induction time. When I say the induction time, this is the time when I'm saying that there is no change happens in the SHG intensity. All right. So this was the perfect time. We saw that even we are not changing any concentration, we are getting some kind of dynamical changes. So what we have done is that we have done in situ real-time dynamics of these samples, because it provides us the opportunity to study simultaneously the diffusion and denaturation process of the protein molecules, right? So in the real-time monitoring of the intensity, so what I have done is that I have chosen two intensities. One of them was the methyl groups coming from the protein molecules. Another one I have chosen is the frequency, which was the characteristic of the water molecule in here. So whatever changes will happen in the protein, they will be reflected in terms of this peak, and the changes happening in the water molecules will be reflected by the red peak in here. So we clearly see that for 5 ppm, there was an induction time. So the sample was lying, then it was not having any changes, let's say until 2000 of seconds. And suddenly the protein molecules start to change their orientation. And we very interestingly can see that these changes are also followed by the water molecules. So there is some kind of synchronized changes happening both for the protein molecules and the water molecules for the this specific concentration. Whereas if you see for the 10 ppm, the induction time reduced. So the induction time was a time when there is no change in the SHG intensity. So it reduced to actually half of it. Right. So the observed fast in case of 10 ppm and the slow growth in here is attributed to increase in the population of surface species by diffusion, followed by the rearrangement of the protein molecule. So initially nothing happens, and then protein molecules start to diffuse toward the interface and they start to show up at the interface. Once they reach in there, they start to unfold by getting exposure to the electronegative oxygen species. And that is what is the second part of the curve in here. So in one line, I can say that the conformational changes of bovine hemoglobin molecule, they synchronize with alteration in the hydrogen bonding coordination of the interfacial water molecules. And just to make sure that this is not happening with just with the SFG, what we have done, that we have done the surface tension measurements for the same. And very clearly, it is visible in here in the first spectrum, the decrease in surface tension. So the standard value of the surface tension of pure water is 72 millinewton per meter, right? So what happened is that it was in here for the Princeton one, we started to increase the concentration of protein molecules and we see that the surface tension is getting reduced. Right, so this suggests that the change in surface hydrophobicity of the surface, right? Because surface tension gives you the change which is happening in terms of the hydrophobicity of the molecules. Now it also we have also investigated the SFG experiment in terms of the conformational changes of the methyl stretching mode as a function of the concentration. So here what we have done is that we varied the concentrations. In here, we have chosen 3 specific concentration, and what we have done is that we took these measurements as a function of the time. And when we clearly see that, remember that during the SFG measurements, which was in here, you see that there was an induction of 2000 seconds for 5 ppm, and the induction time for 10 ppm was the 1000 seconds. You can clearly see in surface tension measurement also that for the 5 ppm, the induction time is still like around 2500 or seconds. And in case of 10 ppm, it's around 1000 seconds. So the induction time is perfectly matching with the SFG measurements. Okay, fine. So this in a way actually validate our SFG findings in here. So this is the molecular model. I thought that when I was talking, I should present it, that what happened is that this is the air and water interface. And as soon as we put hemoglobin molecules, so these red and white dots in here, this is representing a water molecule, where the white ones or gray balls are the hydrogen atoms and the red one is the oxygen atom. This is water molecule in over here. And this kind of ribbon kind of structure, I am representing it as a bovine hemoglobin molecule. What happens is that after a while, this native protein, they start to diffuse towards the interface in here, and it adsorb in here. And it start to denaturate or unfold, which is the same thing, by getting exposure to the electronegative oxygen species present in the air. And we can clearly see it disrupt the water structure. And it also changes its molecular conformation at the interface. So I would say that overall, the extent of conformational changes in bovine hemoglobin at air-water interface is governed by the surface coverage, and low surface coverage result in the substantial unfolding of the hemoglobin. The synchronized changes in CH and OH stretch regions with time clearly indicate the dynamical changes in water network during the intrinsic protein conformational changes. So it's very interesting to notice that how the conformational changes are very well synchronized both in terms of the water molecules and the protein molecules. All right, so here comes the second part of my talk. And you can see that I am representing a kind of dinner lunch plate in here. And for vegetarian people should excuse me. And for the non-vegetarian people, these are the tasty mussels, which is one of the famous seafood. They are not actually anymore for the dinner. So their specific properties, what people or scientists find out that mussels actually withstand powerful currents, and they can also withstand the forceful waves. So they are being detected, or they can be found at the seasides. And they attach themselves to the rocks using clusters of thin, surprisingly hard bisos threads. They can have adhesion to the rough and wet surfaces. So you can see they are having some kind of threads, and then they make sure that they adhere on some kind of surface, let's say on the rocks. These threads in here owe their adhesion power, which is nothing but this bisos. And this is the secret behind the adhesion mechanism. And this bisos is actually an amino acid group called dihydroxyphenylalanine. And from now here on, I will say it as a DOPA, which clings to the surface by performing a series of molecular operations, including the hydrogen bonds and hydrophobic and electrostatic interactions. And this is how the chemical structure of DOPA molecule or dopamine molecule looks like. It's a kind of neurotransmitter. And there was an article in, I think, 2010, which actually have shown the potential of this mucel, which can be applied in chemistry, in material sciences, in cell or surface interaction, in biosensing applications. All right. So what I have done is that I thought that it looks interesting. I should also try it. What I have done that using this dopamine, I have used the concept of molecularly imprinted polymers. So before I will go again to the dopamine, let me very briefly say about the molecularly imprinted polymers. So these polymers are actually kind of recognition element. If you take any sensor element or any sensor system, it consists of mainly two parts. One is the recognition element and other is the transducer. So this recognition system or this recognition element was conventionally before the biological molecules, which suffers from the fact that if they are being placed in their non-native environment, they actually degrade. So, and also they are not very widely or versatile for the detection of different kind of molecules. And this transducer function is to change one kind of information into the other kind of information, or one kind of signal into the other kind of signal. So people thought of not having biological molecules as recognition element, but what they have thought that, okay, let's synthesize something which is very similar to biological receptors or recognition elements. What they have done is that they took the functional monomer unit in here and they took some template. So this is the molecule which we are interested in to sense for. And this is the monomer cross-linker in here. And after the polymerization, this will have some kind of shape in here. It will have functional monomer, and it will also have template molecule, cross-linker and all. After the polymerization, we do some kind of chemical extraction of this template molecule by simply washing. When this template molecule is being removed, it leaves the cavities which are complementary both in shape and size of the template molecule. So let's say if it's a circle, it will have a cavity made out of the circle. Very much similar to the eye products, where if you have an imprint of a half-eaten or a bite-eaten apple, you cannot fit a banana to it, right? So this is the very concept of the molecularly imprinted polymers. And the reason that they are getting popular is because they are more stable in comparison to the biological recognition element, as I already pointed out. And they are easy to produce and integrate with the standard industrial fabrication steps. Anyway, so now coming back to the dopamine, which I have used as a material, it has shown that it can be coated simply by putting any object which you want to coat in the solution of the dopamine. And then after a while, depending on how many hours you are keeping it inside, it forms some kind of thin films on that object. And this particular author Lee et al., they have shown, let's say, more than 10 or 15 kind of object onto which dopamine can be coated. So this is the XPS spectra of 20, like XPS spectrum. And they have taken the substrate signal in here. So it can essentially be coated on gold, silver, platinum, normal glass slides and all. And some of them, they cannot be coated. But since we have variety of materials which it can coat, there is a possibility to do more research. And yeah, this I have already covered in the first part of my talk, so I won't be repeating it again in here. So what I have done is that I simply took a glass slide. I thought, let's try to coat it in here and see that if I can present some kind of sensing surfaces. So what I have done is that I took a dopamine initiator and dopamine molecule, and as an initiator, pH should be adjusted to between 7.5 to 8.5. So basically, the solution should be an alkaline one, which actually initiate the polymerization process. And at the same time, I also placed the template molecule. So it was, this is just a schematic representation. So that is what I was expecting. It will go and sit on the slide ideally in this fashion, and I can remove it. It will leave the cavities, which is the principle of MIP, right? So this is a surface where I have polymerized in the presence of template molecule is going to term as MIP surface. Whereas I have also prepared a reference sample. In here, no template molecule is being placed when the polymerization was taking place. This surface is going to be termed as the MIP surface. And this is simply by putting the glass slide into the solution of the dopamine. So these are some initial results. So polydopamine thin films, they were fabricated with the deposition hours of 3 hours, 6 hours, and 24 hours in here. You can clearly see there is a change in color from light to dark brown, very evident, and is reflecting that while increasing the thickness of the film with the deposition hours. Additionally, the morphological measurements are done using the scanning electron microscopy, and we can see there is a formation of nanoparticles at the surfaces even when we have used the vertical orientation of the sample while the film was getting deposited. But it has been shown very recently in some articles that these nanoparticles are actually good. They have potential in for the antibacterial properties. Anyway, so we have also done some atomic force microscopy measurements to see the thickness of the deposited films. What we have done is that we have done AFM measurements in contact mode. First, we made a fine scratch on the sample with, let's say, a tweezer. This is a hand-on practice how sharp edge you can make. And the measurement was done perpendicular to the scratched line. The AFM images, which are shown in here, are for the 3 hours, 6 hours, and 24 hours of deposition time. And they give the thickness value of around 9 nanometer in here, around 15 nanometer for the B sample, and for the 24 hours, we got the thickness of around 55 nanometers in here, which is in accordance with the literature reported by the Lee et al. So far, so good. I thought things are working out and as much in accordance with the literature. So I thought of, okay, let's do some UV visible absorption profile if I can find any discrimination between the MIP surface and the NIP surface. And you can see clearly in here, using this UV visible absorption spectra, it did not show any significant difference in absorbance of MIP and NIP surfaces for in here. All right, so tada, this was the benefit of the SFG technique. So what I have done is that I have taken all the sample surfaces. I have thought of putting it under the SFG spectroscopy, which is supposed to be the interface sensitive, whereas the UV visible one, it was the bulk sensitive one. I mean, there is always a surface contribution in there as well, but that is not being resolved in those spectrum. So in here, in this slide, what I am showing is that the line profile comparison of the NIP surface. So this is the surface where I have just used dopamine molecule, which polymerized and gave us some nice brown films if we see the naked eyes in here. So in case of non-imprinted films, the prominent peak observed at 2854 in here, and the peak, another peak at 2878, were assigned to the symmetric stretching modes of the non-protonated and protonated ethyl amine groups. These amine groups in here. And since they lose one hydrogen atom, so that peak is also assigned in here. So this is being done for the different polymerization time in here. So far it's good. We can clearly see that with increase in the polymerization time, the increase of the protonated ethyl amine peak, which is this peak in here, is getting increased. Right? This can be due to the decrease in the non-partially polymerized units. Further, the polymerization time of 24 hours clearly depicts the minimal unit or unpartially polymerized DA unit, suggesting that the complete polymerization, which is also reported in literature, that after 24 hours, the polymerization is complete and we cannot actually essentially grow a film of more than 50 nanometers. And this is the spectrum on the right hand side at the bottom I'm representing in here. This is nothing but the fitted spectra to convince the audience, like for their visual guidance, that how I have resolved those peaks which I was talking in terms of non-protonated and protonated ethyl species in here. Okay, and what I have done, that I have taken this spectrum, which is the template molecule. So this time I have just deposited hemoglobin, and I clearly from this slide, it looks a little crowded, but from this slide I clearly want to say that the species which are the molecular footprint of hemoglobin are very well presented in here, and I have already designated them in the first part of the project. This is the third reference sample in which the polymerization of dopamine in presence of template molecule was done. So we can clearly see in here, we observed a clear indication for the presence of bovine hemoglobin in here. In addition to the host polymer of the MIP thin film, the molecular feature at 2854 in here and the feature at 2874 in here, if I'm going to represent it in here, it won't be looking that clear. So that for the guidance of audience, a fitted spectrum is placed at the right hand side bottom of this slide in here. So you can see the PZ peak designation, which I am representing here. Since I have already pointed out that hemoglobin is made of alpha and beta chains, which is abundant in the methyl and aliphatic and aromatic species, all the fingerprints regions are being clearly shown or being clearly confirmed in here. Right. The presence of template molecule during the polymerization of dopamine, it alters the molecular behavior of polydopamine films due to the intermolecular interactions. Now, these template molecules are making some kind of intermolecular interactions, and this is what it is changing the spectral features in here. Subsequently, the increase in the polymerization time from 3 hours to 24 hours leads to the decrease into the peak ratio in here, right? Thus, it indicates that 3 hours and 6 hours of polymerization have higher number of surface species, which are clearly visible in here, if we are interested in the template molecules, in comparison to the 9 hours and 24 hours. So in consideration of the molecular size, which is around 5.5 nanometer for the hemoglobin, and more number of SFG active species present in the case of 3 hours and 6 hours, the deposition time of 6 hours was chosen for the further steps to create cavities and rewinding of template molecules. So in here, what has been done, that these samples were uh, actually washed to remove the template molecules using some chemical methods. So now the hemoglobin molecule should go off. So the first spectrum at the bottom is very much the same, which I was showing in the previous slides, is for the 6 hours. And since it was getting washed, we can clearly see there is an increase in this species in here, which is suggesting now the hemoglobin is losing from the surface. And let me clearly point out that so far the efficiency of MIP surfaces has been achieved to around 40%. And we cannot achieve more than that because we always have to find the optimum for the washing steps, that how much we should actually remove or extract the template molecule so that we will not be destroying the molecular cavities. So that is where somewhere here we stopped in here. And we see that there is absence of hemoglobin peak significantly if we compare it with the 6-hour peak in here. So this species increasing in here clearly saying that now we are more or less having dopamine left on the interface in comparison to the 6-hour MIP intact surface. Okay. Once we got some such kind of interface or the surface where we were having absence of hemoglobin molecule, what we have done that we have done the rebinding of template molecules. So now we got the cavities. We thought, okay, let's see that how much hemoglobin can be bound to these interfaces. And surprisingly, we clearly saw an indication. So black peak in here is for the MIP with cavities, which was having less intensity of this peak in here. If you compare the ratio between these two peaks in the black curve, was less. And as soon as we introduce the hemoglobin molecules in here, and let me tell you in before I concluded, that rebinding was done for almost 20 minutes. And after the rebinding was done, the surface was taken out and washed off to remove any loosely bound template molecule. And we clearly see that even after washing it with the distilled water or any standard chemical solutions, we clearly see that the hemoglobin molecules bind with molecular interaction with this cavity carrying surface in here, clearly suggesting that we got the rebinding of template molecule in this one. So to conclude this part of my talk, I would say that the results I have shown for the second part of my talk, that the polymerization mechanism of the dopamine confirm the molecular footprints and intermediate polymerization species, so which clearly suggests that this is good for the immobilization strategies. Successful imprinted matrix formation was done with molecular conformation of cavity, which can be characterized which cannot be characterized using conventional investigation. So as I have shown that when I put my samples using UV visible, I could not manage to find any clear distinction between the MIP surface and the NIP surface. Of course, I also have to do some more work. Instead of having hemoglobin, I can take any other protein molecules, let's say bovine serum albumin, and then see that how my cavities will respond to that. All right. With that, I would like to acknowledge different funding sources so far for achieving this result. So I also would like to acknowledge the Nonlinear Light Scattering and Biophotonics Lab, which is existing in IIT Ropar in the Department of Physics. And this is the funding which Sabdeep Madam was talking about. I recently got it. And this is about how I can try to utilize the non-machining waste and all the polymer waste to make useful products. And of course, I also would like to thank the Punjab Engineering College. And with that, I also thank all the participants who very patiently listened to my talk. Yeah, thank you very much.
Thank you so much, ma'am. for such an informative, detailed, and interesting talk, which was a blend of biology, chemistry, and physics, of course. So I'm sure that participants would have introduced some ideas for their future research. Like it was a very, very, very good talk. So any questions, participants? If you have any question, please unmute your mic and you can ask, or you can write in the chat box. So I think there is no question, but I have a like not question. I have some like curiosity that are you like seeing in future that what could be their potential uses like application part?
You mean the first part or the second part of the talk?
Maybe both or maybe any of those.
Yeah. So the one which I was talking about, the Mussel inspired research, right? So the recognition of hemoglobin, which is a kind of protein, right?
जी.
Protein sensing is very much demand when
Okay.
you talk about the protein extractions in the biological fields, right? So that is
Okay.
one of the application. And sometimes this I have demonstrated for the protein molecules. But sometimes in environment or anywhere you want to detect some species which are not desired in some specific kind of confined space, right?
Okay.
There also you need some kind of sensing surfaces, so they can be applicable in there too.
Okay. Okay.
Yeah, and
ठीक है.
regarding the okay, yeah, regarding the first part of the talk, which was about the absorption of hemoglobin at air water interface, that was actually coming in from the remember on the first slide or the second slide, I have shown the uh, molecular cell membrane, right?
Yeah.
Yeah, so if we will get the molecular information on the absorption mechanism, having the basic science understanding, this can be applicable in understanding the cell signaling process in the cell membrane. Yeah.
Okay. Wow.
Can be again useful in drug delivery and stuff.
Okay. Okay. Thank you, ma'am. And so like very interesting talk once again. Thanks a lot.
So for the participant who might not be asking some questions, this is my email ID. I posted in chat box, and also it might be visible on the last slide of my talk. So you can always
Yeah.
write me an email regarding any query or anything.
Yeah. Thank you, ma'am. And it would be like if you can share your slides with us, it would be very nice.
Yeah, thanks, sure. Okay, thank you
Thank
very
you.
much.
Thank you. Okay, ma'am.
Thank you.
Thank you, everyone.
Okay. So, dear participants, hello.
Ma'am,
Our next session will be on 3:30 sharp. So please be there at 3:15. We will be admitting participants up to 3:45 maximum. After that, we will
no,
not
ma'am,
accept.
up to 3:30, that disturbs a lot, actually.
Yeah, fine, up to 3:30. So start at 3:15 and we will be accepting up to 3:30 only.
Please share this information with your colleagues and friends who are registered for this course. Dr.
Okay,
Shilpi?
so see you.
Yes,
Shilpi ma'am.
yes, yes,
Hi, Dr. Shilpi.
yes.
Hi, the way of delivering the talk was very appreciable.
Okay, I appreciate. Thank you.
Actually,
Yeah. Okay, then I should
Okay.
leave the session now, I
okay,
guess.
done. Done, done,
Yeah,
yeah.
ma'am.
Yeah.
Bye.
Thank you.
Okay. So goodbye everyone for the time being, and please be there at 3:15, and we will not accept any
We
Google
will not
Meet.
allow anyone to join after 3:30.
Yeah, after 3:30.
And ma'am, certificates. Certificates will be strictly issued to only those participants who will attend at least 80% of the attended sessions. Link will be shared soon.
Thank you.
आ जाओ मैम.